APC's analytical labs are equipped with state-of-the-art equipment for the analysis of small molecule APIs and biopharmaceuticals, including viral vectors and gene therapies. We have the capability to perform a wide range of both liquid and gas-based chromatographic techniques, either in situ for continuous process development or the more traditional offline analysis. Our complete suite of solid-state equipment includes DSC, TGA, XRD, SCM and a full range of PSD equipment offered by Malvern Analytics. We utilise techniques for the determination of protein titer, aggregation, product-related impurities, glycosylation profiling and peptide mapping. Process-related impurities such as HCP, residual host cell DNA and residual protein A are determined by plate-based assays. We offer a wide range of cell-based bio-assays for potency determination. We develop, transfer and validate methods for all types of analysis.